Right. Good afternoon, everybody. Welcome. I think probably know a lot of people in the room, but if you don't know who I am, I'm Steve Darby. I'm the Associate Dean for Research in FELS, Faculty of Environmental and Life Sciences, and it's my pleasure to kind of, I guess, be hosting the event on behalf of the faculty. I'm not going to say very much at all other than to do what I normally do and say that inaugural lectures, we think, I certainly think, are a really important part of the rhythm of faculty life. I've really enjoyed attending all of them across the faculty, and I'm particularly looking forward to Jon's talk today. They are an important part of the fabric of what we do in the faculty, and it's nice to see people here late in the afternoon, looking forward to the event as well. So I really hope that you enjoy it. You're all very welcome, and I hope that you get a lot out of the talk. I'm sure that you will. What I'm going to do is hand over to Chris Horton as Jon's head of school, and give as it were, the formal introduction to him. So thanks, Chris, for doing this.
Thank you. Thanks everyone for coming. Great to see faces on a sunny afternoon in Ocean Sciences Week. So I'm really pleased that you come here. It's great to see some old faces and new faces as well. It's kind of a privilege to be able to introduce Jon to you all, for those of you not so familiar with his background. Jon's been exploring deep ocean for over the past 3 decades, including the use of human-occupied vehicles, remotely operated vehicles, and leading expeditions aboard research ships, discovering new species of deep-sea animals for over 3 decades, as I say. Jon's career started here in Southampton, really, doing a PhD with Paul Tyler, who's in the audience, on the ecology of deep-sea hydrothermal vents from 1994 to 1998. He then stayed on in a number of different roles, eventually becoming a teaching fellow within the school, and really contributing to sort of excellent delivery of deep sea oceanography to many students during that time. Jon became a lecturer in 2003, I think. 2007, thanks, Jon. A senior lecturer from 2013, and became a professor of deep sea biology and science communication in 2022. Jon currently sits on the advisory board of the Global Deep Ocean Stewardship Initiative, so that's a huge international role, really bringing deep ocean issues to the forefront of everyone's thinking. And I'm sure you're going to talk a little bit about that this afternoon, the importance of those roles as we think about how we're going to exploit the deep ocean. Jon's also served as a former associate of the Natural History Museum, and had the role of a National Geographic Explorer. I think Jon's other talent really is in science communication, and since 2006, Jon co-founded a company called SciConnect with colleagues, and that's really been about training new generations of scientists in public speaking, interacting with the media, writing stories for the media. And I know that through various interactions with doctoral training programs and landscape awards around the country, the influence of that organisation has been pretty huge. I won't delay it anymore. I'll just finish up by saying that Jon also holds Royal Society of Biology Science Communicator Award, and in 2007 was awarded the British Science Association Charles Lyell Award and Lecture for Environmental Science. So without further ado, I'll hand over to Jon, who I'm sure will give us a very entertaining talk this afternoon.
Great, thank you, Chris. Thank you, Steve. Good afternoon, everyone. And yes, so when I was little, I had very blonde hair, and I was cross about that. I wanted dark hair so I could be like Mr. Spock. Mr. Spock was my hero because he was different, and his job seemed to involve knowing things or finding things out if he didn't already know them. And that job, of course, was famously to explore strange new worlds, to seek out new life, and to boldly go where no one has gone before. And I really like the sound of that as a job. But where in the late 20th century could I find a job that was in any way somehow like that? Well, the answer that I found was as a biologist working in the deep ocean. And you actually get that strange new worlds evoked here in this quote from Charles Wyville Thomson, who led the famous voyage of HMS Challenger in the 1870s. The first voyage really to explore deep sea, to explore deep sea life around the world. And as you came into this building through the main entrance, you would come past a replica of the figurehead of HMS Challenger. But these strange new worlds, they're just starting to be opened up in the deep ocean. So that's what I'd like to talk to you about this afternoon, is kind of my journey in deep sea biology. And the overall aim of my research, very simply, is to understand what lives where and why in the deep ocean. And as I'll share with you, that has involved piecing together a global jigsaw puzzle of deep sea life. Now, I called this Beyond the Blue Planet as part of the title because the blue appearance of our planet from space, that famous blue marble, that blue comes from sunlight reflecting off actually a thin surface veneer of the ocean. Most of the ocean is deeper than the sunlight goes, deeper than the blue, beyond the blue. So, ours isn't just an ocean planet, it is a dark, deep ocean planet, at least relative to our human scale. So the statistics I like to use for that is, the average depth of the ocean is about 3,400 meters, or look at it another way, more than half our world is covered by water that's more than 2 miles deep. And I think most importantly is the realization that that deep ocean is not one environment or habitat. It is just as rich and varied as the world above the waves. So there are vast, muddy abyssal plains, there are rocky mid-ocean ridges and seamounts, there are some places where the life is incredibly abundant. There's that vast mid-water kind of inner space habitat in the deep ocean. Lots of different habitats, just like the land. Okay, much bigger than all the world's land masses. So that's the deep ocean that we're exploring. We talk about deep ocean starting at 200 meters. So it's the majority of the ocean by area and volume, of course. Now, growing up, I was fascinated by animals. I was inspired, of course, like many of us, by TV natural history documentaries, David Attenborough series over the years. And so I took a degree in zoology at the University of Sheffield. And then one day when I was walking through the library there, I saw a volume up on the shelf, and it had a picture on it of some very strange looking worms. And I saw this and I thought, hang on, I'm doing a zoology degree. I'm not even sure I can put, what group of animals those worms belong to. So I fetched this volume down, and it was a special edition of the journal Deep Sea Research, full of research papers about discoveries at deep sea hydrothermal vents, one of the habitats we get in the deep ocean, which had only been discovered in the late 1970s. By the time this volume was published, that was just over 10 years that these things had been studied. And I was totally blown away by this. It wasn't covered in any of my lectures at the time, because it's still a relatively new discovery, and it kind of overturned the rules I'd been taught at school in biology about how ecosystems are supplied with energy. Because these deep sea vents are like hot springs on the ocean floor, very much like the geysers you get in Iceland or Yellowstone, but at the bottom of the ocean. And rather than erupting steam, they erupt a hot mineral-rich fluid. And the minerals dissolved in that hot fluid, in turn, are an energy source for microbes, and then the microbes are a food source for animals, and you get these colonies of astoundingly abundant deep sea animals thriving island-like around this set of hot springs. So those are deep sea vents. And I was just utterly captivated by this. For the rest of my undergraduate degree, whenever I could twist a coursework assignment to make it somehow about deep sea vents so I could spend more time finding out more about them, I did. And at the end of my degree, I got some very good advice from my tutor, which was, don't do a PhD. With the caveat, unless it's on a subject about which you are utterly passionate and prepared to give blood, sweat and tears. And I thought, well, there's only one topic that fulfills those criteria for me. So I spent a little bit of time working and saving some money, and then I came to Southampton to do the MSc in Oceanography, to become, get some marine science background, having done zoology before. And that is where I met Paul Tyler. So Paul has spent more time directly observing life in the deep ocean, I think, than any biologist in British history beforehand, working with human occupied vehicles. Paul literally wrote the book on deep sea biology. And during my MSc, Paul got the first funded research grant in the UK to investigate the biology of deep sea hydrothermal vents. So that had funding for a research assistant, which was then me, and I was able to complete a PhD as part of that. And so, Paul, I am one of many students who've gone on to work in this field around the world who can honestly say, I wouldn't be standing here today if it wasn't for you. We've been shipmates on many expeditions together. This is a picture from one of my favourites. This is raising a toast at Shackleton's grave on South Georgia. And I was counting up the other day, we've actually been co-authors on 24 research papers together over 29 years, starting with my master's project. And then last year, Christopher McNeil was a master's student, published his project last year, and we were able to tempt Paul out of retirement to train the student in how to do reproductive histology in the lab. So Paul, I'm so delighted that you're here. I remember being in the audience as a PhD student at your inaugural. Okay, so those deep sea vents, island-like is the analogy I like to use. They are dotted along the global network of undersea volcanic rifts where the plates of the ocean crust are rifting apart as they ride on convection cells in the mantle. And you get these set of hot springs, colony of deep sea life around it, some distance to the next. They are island-like. And so it's very much like the kind of Victorian naturalists who explored life on islands above the waves to understand more about how species evolve and disperse. Same kind of thing. We can use deep sea vents, almost like a model system, to do that, to understand how things disperse and evolve in the deep ocean. So this painting is an imagining of Beagle with Charles Darwin actually arriving at the Galapagos. So there's kind of that theme, if you like, running through this. So since they were discovered in the late 1970s, they've been sort of steadily being explored. And if you look at the numbers that have been visually surveyed on the seabed, it started in the late 1970s. By 2010, it was more than 200 had been visually surveyed. And around that same time, the number of new animal species that had been described from those deep sea vents went from 0 to more than 400. So when you look at these graphs, you might think, oh, so if you find a new set of deep sea vents, on average, you might expect to find 2 new animal species. But it doesn't actually work like that, because that deep sea life is actually partitioned into what we call biogeographic provinces. And these are the puzzle pieces in our global jigsaw. So what that means is all the deep sea vents within a particular area, like this area in the North Atlantic here, they share a common pool of species, just kind of similar species at all the vents in that area. Then you move to a different region, and all the vents in that area have a different set of species occupying those vents. And this was the global jigsaw puzzle at the start of my PhD. There were only 4 puzzle pieces, 4 areas where people had explored life at those deep sea vents. And so for my PhD, I worked on the patterns of life within 2 of them, in the North Atlantic and the Northeast Pacific. And looking at in detail at patterns of life you get around the vents, but not really the bigger puzzle, not putting new puzzle pieces onto the map there. Now, during my PhD, I also started my parallel career in science communication, because there were some popular science writing competitions back then. The Daily Telegraph ran one, and I was runner-up in that. New Scientist ran one, and I won that. And that then led to an opportunity after my PhD to become an intern at New Scientist magazine. And then I actually stayed on there as a reporter and then became assistant news editor. And I was there for a couple of years after my PhD. And subsequent to that, I carried on working as a freelance science journalist, writing for New Scientist and places like Nature as well. So that's kind of a parallel career that started around the same time. But I still wanted to do things in the deep sea. So I was lucky enough to have the opportunity to come back to Southampton to take up a teaching fellow position, as Chris mentioned, working in School of Ocean Earth Science, very much focused in fieldwork teaching, which is something we're very strong on, I think, here in Southampton. And so the foundation of my academic career is very much in teaching. And I think the things I'm proudest of, really, are in education. Actually, my most cited research paper, by a big margin, is in education rather than deep sea biology, as it happens. But being a teaching fellow was a little bit of a catch-22 when it comes to doing research. Because to get a research grant to do deep sea ecology, with a research expedition, well, those take several years to schedule, and these kinds of positions, early in your career are often, short-term temporary contracts, and you need to be able to prove you've going to have a job if a research grant is funded. Of course, therefore, to be able to put in those grants, you need an open-ended position. To get an open-ended position, you need a decent set of research papers. To get those research papers, you really need to be working on funded research projects. So you're kind of stuck in this situation. I was fortunate because I had opportunities to join deep sea research expeditions, invitations from colleagues in the US and elsewhere, and also people offering to send me samples to analyse and so on. So I actually funded my own research with my income from popular science writing, which also kind of saw me through that precarity. And that eventually meant I had enough papers I could secure an open-ended position, and then I could be an investigator on grant proposals. So this was the jigsaw puzzle at that point. Not much had changed since my PhD. There had been one additional puzzle piece in the central Indian Ocean, where people had started to explore deep sea vents there. So since then, really, most of my career, a big theme of it has been finding additional puzzle pieces, filling in the gaps on the map. And that's what I want to take you through now. So starting with a big puzzle piece down here in the Southern Ocean. And this was on a big grant that Paul put together and led with colleagues around the UK, which I was able to join as a co-investigator. And this is an interesting puzzle piece to target. So the question here is, okay, the life that we find at any deep sea vents in this region, will it be like the life in the Atlantic, to which it's kind of connected today? Will it be like the life we've seen in the Pacific, because there's an enormous ocean current that sweeps through here, in this case it goes around the Antarctic? Will it be completely different? Okay, because it could be isolated and so on. So that's really what broad terms, that's what we set out to answer. 6-year program, 4 expeditions with research ships and vehicles and so on. And you can see, strange new worlds kind of being evoked in some of the media coverage of what we discovered. So here are some of the deep sea vents that we found down in the Southern Ocean. This is 2,400 meters deep, and these are these mineral spires, vent chimneys as we call them. Each of these that one is about 6 meters high. That one's a little bit smaller, but that one's about 6 meters high there. And as you can see, particularly this one, this is probably my favorite anywhere in the world. We call this one car wash because it has these 2 spires that are covered in stalked barnacles. It looks like a car wash. Just covered in animals. Stalked barnacles, these are crabs here, all this brown stuff, these are snails. Every bit of that surface covered in life, 2,400 meters deep in the Antarctic. Phenomenal, absolutely phenomenal. And in fact, I've got some postcards left over from a public engagement project. If anyone wants a memento with that image, you're very welcome to come and help yourselves to those. Now, that's also not a single picture, okay? Because if you were to back, in this case, our remotely operated vehicle away so you could kind of see the whole structure, it would fade into the darkness and you wouldn't be able to see it. So this is actually a result of refinement of a technique I'd started to use a bit and develop in my PhD, but with some much better technology now. Some very precise flying with our ROV, close in with a forward-looking camera, basically scanning the structure with the camera, and then taking individual frame grabs, stitching them together. So that whole image is about 800 frame grabs stitched together. And that means we can zoom all the way in and we can count limpets on the backs of crabs and, do that kind of thing. We've got fantastic detail to analyse the patterns of life. And we do that with things that are upstanding, like vent chimneys. We also have a downward-looking camera. And again, we kind of mow the lawn with the deep sea vehicle, downward-looking camera, and then recreate that seafloor digitally, covering, a couple of hundred square meters. And then we've got a digital representation. We can do what the ecologists do on the rocky shore, where they go and they chuck quadrats about. And that kind of, we kind of do that in a way that we can't really in the deep ocean. So from that, we're able to piece together patterns of life. And there's a distinct zonation of species as you move away from a deep sea vent. Very much similar to the zonation of life you get on a rocky shore as you kind of go down the shore. So we're able to piece these things together, and all of the species at these vents new. Okay, not related particularly to the Atlantic or the Pacific. They're all new at species level, not seen before. More than 30 new species from these vents. And here's one that's really abundant close into the vents themselves, okay? Kiwa tyleri, that was the name of the skin when it was properly described, nicknamed the Hoff crab by PhD student I co-supervised, Nikolai Roterman, because it has a hairy chest, like the Baywatch actor David Hasselhoff. This got into the media and is really stuck and got some traction. But these crabs live in the warm vent fluids where there's dissolved hydrogen sulfide, which fuels bacteria that grow on their hairy undersides. And they scrape the bacteria off their chest hairs and eat them. And that is how they survive. In huge abundance. There are these piles and piles of crabs that you can see here. Now, this is where the crabs are feeding and so on. And also, this is where the males and females are mingling. Okay, and another pattern of life we predicted, we're able to define, is the females then, once they've spawned their eggs, the males have fertilized them, they then brood their embryos tucked under their carapace, like a lot of crabs do. This is not a good place to do that because the vent fluid is a bit toxic and there's a lot of argy-bargy with all those crabs. So the females then crawl away from the vents and they're dotted around the periphery individually. And they can't feed when they're out there and they're just waiting for those embryos to develop and hatch. And then they come back in. The males, in the meantime, stay here, stuffing their faces. Okay. And consequently, the males are about 3 times the size of the females, which is very unusual for crab-like animals. So this was a really interesting species because it had this interesting life story. We had really good video footage of it. We pieced together a life story of it. And we have really good video. And of course, those are the kinds of things that natural history filmmakers want, so they can tell a story and they can tell it visually. So this led to a relationship with the BBC Natural History Unit, who featured the Hoff crab in a documentary series that was on BBC Two in 2014, I think it was. And then that led to working with them on other projects. So thanks to the Hoff crab, I became a science advisor for the Natural History Unit while they were producing Blue Planet 2. So I worked with them for a couple of years on helping find stories, deep sea stories for that program, culminating in the opportunity to join their expedition to film in the Antarctic, where we made the first dives in a human occupied vehicle to reach 1,000 meters deep in the Antarctic. We weren't visiting deep sea vents, we were filming other things, but I wouldn't have got there if not for the Hoff crab. So that was something that kind of came out of that unexpectedly. That Southern Ocean puzzle piece had so many discoveries in it, as well as the vents that I've shown you, which are kind of like other deep sea vents around the world, but with different marine life here. We also found a very different kind of hydrothermal vent. And we found that at the bottom of the South Sandwich Islands. Okay, so here on this map, the black blobs here are the actual South Sandwich Islands. These are the southernmost set. And then there are also seamounts, undersea volcanoes that haven't grown quite enough to break the surface and become islands. And there's one here called Kemp Seamount, and it's seamounts are also really interesting deep sea habitat. So on our first expedition, we went and towed a camera across that, and we had a very exciting dive looking at marine life on that. Then biology team went to get some rest, go to bed, left the geophysicists in charge of the ship to do whatever they wanted with, and they decided this was the best map of the area we had at the time. And it didn't indicate anything interesting near Kemp Seamount, because this is derived from satellite data. Ship-based sonar gives you a much more detailed picture of the seabed. So they decided to do some mapping with the ship's sonar around Kemp Seamount, which hadn't been mapped before, and they found, this is Kemp Seamount down here, right next to it, just to the northeast, this seafloor caldera, crater-like feature in the seabed. 4 kilometres across, 1.6 kilometres deep. Nobody knew that was there until literally that night they switched on the sonar and found this thing, which I think is astounding. Where else in the world can you trip over something like that people didn't know was there before? So very exciting. And then at the heart of it, there's a little resurgent volcanic cone. And then at the top, on the upper slopes of that volcanic cone, a set of deep sea vents. Very different in chemistry and environment to the other ones. Okay, different chemistry. The structures here are made of elemental sulfur. Not the same minerals as those other vents, and different marine life. And then, even better than that, whilst we were then surveying this discovery, purely by accident, we came across the skeleton of a minke whale nearby. And whale carcasses and whale skeletons on the ocean floor also support other island-like colonies of deep-sea life. In this case, there were 9 new species of animals. And that's the first whale fall studied in situ in the Antarctic, even though there's a lot of whales down there. Because You can't find it. There's no way of detecting these remotely. You literally have to blunder over them by chance with a camera or a deep sea vehicle. So it was a phenomenal project down there, and I really know how those Victorian naturalists must have felt turning up in places like the Galapagos.
2 species in as many minutes.
It really felt like that. So, okay, that's our first puzzle piece. I'll take you to another one now. This one rather warmer, and this kind of overlapped, in the Caribbean. And this is a place we've been targeting for wanting to get to for a while. Again, here the question is, we know what lives at vents here in the North Atlantic, and maybe what we find at vents in the Caribbean will be related to that, because oceanographically they're kind of connected today. But until between 10 to 3 million years ago, there was a seaway, of course, through what is now Isthmus of Panama. That hadn't quite closed off. The deep connection closed about 10 million years ago. So could deep sea life here be related to what we know is in the Eastern Pacific? Or again, could it be different to both? So that's what we set out to find. This was the first grant that I was able to lead as PI and put together, and we got funding to get out there and find the deep sea vents. So there is an undersea volcanic rift in a part of the Caribbean called the Cayman Trough. It's the world's deepest undersea volcanic rift. And we found the world's deepest hydrothermal vents there. So this is the volcanic rift. It runs north to south. It's about 110 kilometers long. We found the deepest vents where you expect to find deep sea vents, at the heart of the volcanic rift, 5,000 meters deep there. But we found another set of deep sea vents in a place where no one would have expected. There is this kind of undersea mountain. It's called Mount Dent. It's not a volcanic seamount. It's actually what's called an oceanic core complex. That's where a block of crust has got pulled up out of the ocean crust as it's spreading and kind of laid over on its side to form a mountain-like feature. And on the upper slopes of Mount Dent, we found another deep sea vent, totally different to others that have been found around the world. So the deepest one you can see here, submarine life. The other one here, 2,300 meters deep, which we named Von Dam vent field after Karen Von Dam, vent geochemist. Very different in chemistry. That mound there is made of talc, literally like talc, So totally different chemistry, mineralogy, different marine life. These sites are only 20 kilometers apart. They're in the same water mass, so they're the same conditions of temperature and salinity, and yet they only have one species shared in common between them. Otherwise, they're totally different in terms of marine life. So this tells us something about what lives where. These are the spires of the deepest vents, again with our scanning technology. This one's about 6 meters high, this complex here. These ones are about 4 meters high. Really slender, look really different to the ones in the Southern Ocean. And of course, all the species are different. And here's the one species that's shared between the two sets of vents. Rubicaris hydrocybe is a new species that we described. And you can see it here, huge abundance of them on those vent chimneys. This is a shrimp, and in some ways similar to that Hoff crab. The shrimp gardens bacteria on its gills and on its mouth parts, scrapes them off and eats them. So similar kind of lifestyle in some ways. And other interesting features, it doesn't have eyes on stalks like shrimp normally do. Instead, you can make them out if they're in the video. These white coloured patches on its back are a light sensing organ that it has on its back, because deep sea vents glow incredibly faintly. The shrimp need to be able to find their way around them. Part of how they do that is they have this very different kind of light sensing system. And we named it Hydesci after the deep sea vehicle that we used to collect the first specimens. So we had 2 expeditions as part of this project. The first was very much reconnaissance, and NERC wouldn't let us take the very expensive ISIS ROV with us because we didn't know if we were going to find the vents. So we had this thing called HIBIS, Hydraulic Benthic Interactive Sampler, which is basically a TV grab. You can maneuver it about, it's got a camera, it's got a grab for kind of getting geological samples, but it's not designed for biological sampling. Normally when we want to collect something like shrimp, we have a thing like an underwater vacuum cleaner that we call a slurp gun. We didn't have one of those on HIBIS. So when we found the vents on the first expedition, we saw the shrimp, we thought, it'd be really great to get some specimens we could start to analyse right now. We were able to bodge together a slurp gun out of bits on the ship. So buckets, duct tape. We took a standalone pump from another bit of equipment, and we managed to get those first specimens. So we gave it that species name. On the second expedition, we had the ROV. We were able to sample much more thoroughly. And one of the things we did was we mapped the vent area in great detail. So this is flying the ROV about 80 meters above the seabed with a scanning sonar on it. And we get this map of the whole area. There's a 100-meter scale. And all these bumps that you can see on the map, they are real. Those are individual boulders you can see on the seafloor. So this is great because we have a map that you can use to find your way around. And consequently, that led to an opportunity to join a Japanese expedition 6 months after our second expedition. They wanted to dive there with their human-occupied vehicle, which could reach those vents at 5,000 meters deep. And I got to go along on the first dive simply because I had the map. But that was a great opportunity. So we were able to work with these guys, get more samples, spend more time investigating that habitat. So there's another puzzle piece. So for another puzzle piece, I mentioned that people had started exploring vents in early 2000s in the central Indian Ocean. The Indian Ocean is interesting because the mid-ocean ridge, the undersea volcanic rift, there's kind of three parts to it in the Indian Ocean. There's a central Indian ridge, there's a southwest Indian ridge, there's a southeast Indian ridge. And the central Indian ridge, where people have seen vents already, different geologically to the southwest Indian ridge. So the question was, well, what would life be like on the southwest Indian ridge? So that was our next puzzle piece to target. And a really good opportunity to do this because Alex Rogers had already had a NERC project funded to look at seamounts along the southwest Indian ridge, actually getting one of our ships to the Indian Ocean with an ROV and all that kind of thing. So I was able to put in for a small grant for what was then called bolt-on ship time, basically to fund 3 days of ship time to divert the ship during Alex's expedition for my little mini expedition to explore a set of deep sea vents that have been detected there, photographed with an AUV by a Chinese expedition in 2008, but they hadn't collected any specimens. They just confirmed where the vents were. So that's what we did. We went out there and we spent 72 hours working at those vents. And I'm very proud of this one because from those 72 hours of field work, we got samples and data that contributed to more than 20 research papers. So NERC should be pretty happy. I think that's good bang for buck. And, you know, it's a good, efficient use of research ships to be able to bolt on, I think, like that. Okay, so what did we find? Here are those vents, 2,800 meters deep here. This is a mineral spar. This is about 4.5 meters tall here. This is the video, is the top of a really spectacular one that we found, 25 meters high, several stories high. This is what we see at the top. In terms of marine life, there's some stalked barnacles. These are mussels. And then those little brown blobs are snails. This is close up. The shimmering water is the warm vent fluid. You can see there are some shrimp. There's something that is very similar to the Hoff crab from the Southern Ocean. And then this heaving mass here, these are snails. And they're very odd looking snails. They've got these very dark shells. And then they've got all this dark stuff on the fleshy foot of the snail as well. And so this is one of the species that's very abundant at these vents. And it's a species called, well, nicknamed the scaly-foot snail. That's its proper name. This is a species that had been found at those central Indian Ocean vents in the early 2000s, but it had never been properly described as a new species. So Chong Chen, who was a PhD student, I co-supervised with Alex Rogers, Chong formally described it as a new species as part of his PhD. And this animal, how does this survive? Well, it doesn't garden bacteria on its body and scrape them off. Instead, it has a bag-like organ attached to its esophagus, and it cultures bacteria inside that and digests them. Okay, so it kind of grows its food in this bag off of its throat, if you like. But what's unusual, why it's called the scaly-foot snail, these things on its fleshy foot, you know, that's not crud or anything. Those are plates of a metal mineral, okay, that are deposited on this snail's foot. And that is the end product of a detoxification mechanism that this snail has. Because where it lives in those vent fluids, there's high dissolved metals. It needs to stay there because that's where the energy source is for the bacteria inside it, for its food. And to cope with the toxicity, it deposits the metals in these mineral plates that it grows on its foot. Okay, so really quite remarkable adaptations. And I'm very proud of this one because it is also the star of a forthcoming in the UK episode of The Octonauts, some of you might be familiar with. Season 5 of The Octonauts hasn't fully broadcast in Britain yet. It has in China. There is an episode, The Octonauts and the Scaly-foot Snail. So it's great to see a species I've had some involvement with making it into that. Okay, so that was Southwest Indian Ocean. Now, looking at the map, there is a really obvious gap right in our own backyard. People had explored life at deep sea vents south of the Azores in the Atlantic, but not between the Azores and Iceland, where the northern Mid-Atlantic ridge runs. So again, this is a place we've been wanting to get to for a while. Bram Merton here at NOC in particular, I wanted to target this, and we had a couple of goes at getting funding. Eventually, we managed to get some funding out there working with some Irish colleagues using eventually the Celtic Explorer Islands research ship and their ROV. We had some funding from National Geographic as well. And we got out there, and Bram had previously detected where we thought there would be a set of deep sea vents out there north of the Azores, and we were able to find them and survey them in the same way. So here are those vent chimneys. These are about 4.5 meters high, this one on the left here. This on the right is the top of, again, a really massive chimney, more than 20 meters high. It was a bit challenging working here, though. This site is at 3,020 to 3,050 meters deep. And it's on a steep slope right on the edge of the Rift Valley of the undersea volcanic rift, not where we were expecting to find the vents, I have to say. And consequently, the ROV, the remotely operated vehicle we have, limited to 3,000 meters. So it was challenging, not in terms of mechanically going a bit deeper because there's a safety factor in these things, but actually running out of cable connecting it to the ship. We had to stretch out all the cable we had, which normally you put plenty out and then the vehicle stays steady while the ship's going up and down in the waves. We had to stretch it all out to try and reach the bottom of these chimneys to survey them, and the whole lot was going up and down with the ship at that point. So it was challenging, but in terms of marine life, same species as the ones found south of the Azores. So sometimes it's exciting when it's all new, but sometimes the big answer is actually it's the same. So that puzzle piece is really just an extension of that other puzzle piece. That's important for our understanding. So finally, the last puzzle piece I've been working on most recently is up in the Arctic. Okay, now the Arctic is also a really interesting place to go because the deep Arctic basin is pretty well isolated from the rest of the world's deep ocean. There's only one deep sea kind of channel connecting it to the rest of the world's deep oceans. And in the past, it's been very different. You know, there was a thick ice sheet up here. It was a freshwater lake, you know, for a while, you know. So the Arctic is really exciting in terms of what will we find there because it's had this very different environmental history. Okay, so this is work I was involved in most recently. We had an expedition in May 2024 as part of the International Ocean Census Programme. And that was on board a Norwegian icebreaker research vessel with a remotely operated vehicle. And we published the main discovery from that actually just before Christmas. Okay. And what that main discovery was, what we found, we were looking at deep sea vents, which I'll come back to in a moment, but we found another kind of island-like habitat in the deep sea we get called cold seeps, very similar to deep sea vents, again, chemically fueled rather than solar powered and so on. And we found what are the world's deepest methane hydrate cold seeps. So this is where a really bizarre substance called methane hydrate, sometimes called methane clathrate or gas hydrate. It's a kind of methane water ice. It's water molecules that form a crystalline lattice and they trap a methane molecule inside them. And you get deposits of this, you know, under the seabed, buried, you know, where there's organic rich sediments and so on. But occasionally it outcrops at the seafloor. And we found an area, a couple of football pitches in size, where there were these mounds. This is where the hydrate is bulging up out of the seafloor, and then as it gets exposed to the seawater, it fizzes away. Okay, and that then produces some structures that are arches, where the hydrate is fizzed away, and then pits. And that releases a plume of methane bubbles that we were able to detect with the ship's sonar that rise for 3,300 meters above the site. That's how we found it, was finding this bubble plume. They don't reach the surface. Those bubbles are coated in hydrate. Okay, and that means that they don't dissolve into the ocean while they rise up through it. Hydrate is only stable under low temperature and high pressure conditions. So the bubbles rise up through the ocean, and then they get to a point where the pressure is less, the temperature gets warmer, they lose that hydrate coating, and then the methane dissolves into the ocean really rapidly. So fortunately, the methane released from this site does not reach the atmosphere and contribute, you know, to a potent greenhouse gas. Nor will it in future, because ocean warming is going to increase the thickness of that warmer layer at the surface, hopefully. So, you know, we don't have to worry about that here. But what was interesting was the marine life around them. So we have the hydrates here. They're covered with what we call the tube worm forest. This is dense sort of tube worms and then other animals, shrimp and so on, living amongst them. And what we showed was the life at these deep seeps was closely related to the life at hydrothermal vents nearby at similar depth, and less related to other methane hydrate seeps elsewhere in the Arctic. The other hydrate seeps about half the depth that people had seen before. So the life here, part of a network of life connected to those hydrothermal vents. And the animals, totally different to the ones we find at vents anywhere else in the world. Here's a few of them. This is one of my favorites. This is a Stauromedusa. That's kind of like an upside down jellyfish on a stalk, lives attached to the seabed. So the International Ocean Census is still analysing these to describe them as new species and give us a more detailed picture of the life there. So that was our jigsaw puzzle to start. Okay, this is the jigsaw puzzle now with those extra pieces filled in. And of course, this is a huge collective team effort in all this kind of research. This is many teams working together on individual expeditions, on different research grants over the years. You know, it takes more than a village to do any of this kind of research, to get out in the deep ocean, all the people supporting you on the ship as a science team as well. And then, of course, within that community, the research students over the years who've contributed to all this work. And as PhD students do, many of them have gone on to make contributions in lots of different sectors, which is what PhDs do. A few of them have continued as well in marine science, and it's been such a pleasure to continue working with them as early career researchers and being co-authors on papers together, you know, right up to today, and to do that as well. So I'm very grateful to all their contributions. I mean, obviously they're the ones that really innovate and drive the research. What does it all mean, though, okay, in terms of this puzzle? Well, I hope I've given you a flavour of how, you know, deep sea life does really vary from puzzle piece to puzzle piece for the most part. And when you actually do that analysis properly, this is the kind of picture you get. So here I've shown the puzzle pieces we've filled in, and I've colour-coded them, so anything that's a different colour is a different biogeographic province. And you can see that some of them are very distinct, they're different colours to anything nearby. Some of them, I mentioned the one in the Atlantic, that's really just now an extension of the one that we knew was to the south. The Indian Ocean, it's a subset of things that are found at the Central Indian Ocean, and it doesn't quite become a province in its own right. But this is the kind of insight we get as to, you know, how species disperse and evolve. Then we can ask ourselves, well, what are the barriers? What makes this different to that, even though they're close by? And then within this, there are some species that conform to these patterns. There are a few species that deviate from. There's a finer detailed picture. So there are some species whose distributions span several different coloured puzzle pieces. And then the further question is why? Why is that species so widespread? In some cases, it may be because they're really good at dispersing. You know, they have a larval stage that's very good at being transported long distances. In some cases, they might not be that great at dispersing, but evolutionarily, they may be really old and they've had plenty of time to get around, even though they're not particularly great at getting around. So then we can understand these things. And what about the species themselves? You know, so lots of new species have been involved in describing over the years in lots of different types of animals: fish, crustaceans, mollusks, worms, echinoderms, starfish as well. So, but a very valid question, and I've even had it from, you know, politicians and so on, is, okay, you found a new type of snail at the bottom of the ocean. So what? Honestly, who cares? What's the big deal? And there are lots of answers to that, and I have one in particular that I like to share when I get that kind of question. Which is relating to what we can learn from these species. So I showed you the Rumicaris hibisci shrimp. One of the features that this has is tiny, tiny little bristle-like structures on some of its appendages, really, really tiny. And they have now inspired a new material from material scientists, a nanofibrous aerogel, and that has really good properties for heat and sound insulation. So a new material for heat and sound insulation inspired by structures on this deep sea shrimp that we found. It's probably going to be used now to make Chinese submarines quieter and harder to detect, but you know, that's how it goes with these kinds of things. Here's another one. I showed you the scalyfoot snail. Now, when it's making those metal plates, it is creating tiny crystals of a mineral called pyrite at room temperature. And the people who make solar panels would love to know how to do that, it turns out. And now a team in Japan has figured out how the snail does it. And normally, the way that people do this for solar panels is it's a very high temperature process using some pretty nasty chemicals. The snail does it at room temperature, and they've recreated this process with off-the-shelf ingredients that are far less environmentally challenging. So this snail is literally teaching us how to make better solar panels. Okay. So those are a few examples from material science. There's then also a whole bunch, of course, in kind of medical science as well. So this is not species I've been involved in or involved in this research at all, but these are just some examples I tend to carry around. There is, for example, a new therapy for some types of prostate cancer that may be resistant, more resistant to existing other therapies. And at the heart of how it works, there's a molecule that makes cancer cells sensitive to light, and so they can be zapped with a laser. That molecule was developed over a period of 20 years and highly modified, but it was originally discovered in deep sea bacteria. Okay, so it came from the deep sea. Then a big health challenge for the future. Unfortunately, by 2050, projections are that antibiotic resistance will kill as many people globally as cancers do. Of course, we need new ways of tackling antibiotic resistance. The deep sea offers potential for that. And then there are other things for other problems that we have, understanding how deep sea, there's a bacteria that can break down polyurethane. Could that be used to help tackle some environmental problems that we have? Now, this is something that I've been interested in for a long time. This was the subject of my popular science article for the Daily Telegraph competition many years ago. So it's been great to kind of come back to that. And through a project funded by DEFRA that we've recently had here and working with colleagues around the UK called DEEPEND, Deep Ocean Resources and Biodiscovery. And that included sending samples off to people that have the analytical know-how to, that we collected in the deep sea, and they're finding new molecules with interesting potential properties. But the thing we did in Southampton was to map all of the molecules that have been found in deep sea organisms that have potentially interesting properties. Where have they come from? Produce a map so far. And this is work led by Lucy Harris. Lucy's now doing a PhD here at Knox, but Lucy was a research associate on the DEEPEND project before that. And we're about to publish this in Scientific Reports. The final stages of review are now being sorted out. So Lucy led this. We mapped all of the records of what marine natural products that have been published from deep sea organisms to look at their distribution in space. A lot of them are from exclusive economic zones of countries, fewer from the high seas areas. We looked at the depth distribution. We looked at their distribution on the tree of life. And there are whole branches of animal life where no one's ever looked for any interesting compounds yet from the deep sea. So it's kind of a roadmap to guide our future discovery. And the analogy I use when I'm talking to politicians is that the deep sea is like a library. Its biodiversity is like a library of the ingenuity of nature. And, we're still learning, reading things in that library. It's a lot we haven't read yet. And of course, this is a depiction of the famous Library of Alexandria, which burnt down, losing much of the knowledge of the ancient world. And of course, this is the context. Okay, this is one of the biggest reasons for showing this potential from deep sea biodiversity is it is very much under threat from human impacts. It's not as remote and so on. So impacts, everything from litter. This is not a picture on a beach. This is 2,300 meters deep in the Cayman Trough. Okay, Stella can and a bin liner, and we found other litter at 5,000 meters deep. There's microplastics you can't see as well, of course, everywhere. Illegal, unregulated fishing. This was an illegal trawler at a seamount in the Indian Ocean that we had a little bit of a conflict with when we wanted to work on that seamount on the Indian Ocean expedition. Climate change, of course, huge impacts on the deep sea in lots of different ways. Okay, changing chemistry, ocean acidification, so on, but in particular deoxygenation, changing the flow of oxygen from polar regions, where it gets the oxygen from the atmosphere down into all the deep sea habitats we've looked at so far. And on the cars, of course, deep sea mining, extraction of those kinds of resources from the deep sea. So, how did it turn out then? That childhood imagining of a job like Mr. Spock. Okay, let's have a quick think. Seek out new life, yes. Go where no one's gone before, that too. But explore strange new worlds, actually no. Because the realization is very much that these deep sea habitats, they may seem remote, they may seem alien, but they are very much connected and part of our world. And it's kind of a paradox of the deep sea. We think of it as dark and remote and alien and so on. But here's another way of looking at it. If the Earth were the size of a football, the depth of the ocean covering it would be about the thickness of a postcard. It is not as remote as we imagine. It is far more connected and vulnerable. So that's been a theme that I have tried to, get across in my parallel science communication career. So we had the MOOC here in Southampton over many years. Well, that was very much a realization we tried to lead the learners through to and get them to have that increased awareness. It's a theme that runs through my popular science books. and through my public engagement and media work as well over the years. But I think there is a bit of a challenge when it comes to deep ocean science communication, because the deep ocean, historically, culturally, mythically, is this dark void space. It's not like the starry night sky. We can go out, look up at the sky, imagine space and what's up there. This is this kind of unknown underworld that we populate then with creations of our imagination. Okay, and we see that in myth, but we see it in popular culture as well. One of the first deep sea science fiction stories, H.G. Wells, okay, called In the Abyss. We see it today in Hollywood. The movie Underwater in 2020, really the same story as H.G. Wells In the Abyss. And of course, perhaps captured in that famous quote from Nietzsche, although he wasn't referring to the abyss of the deep ocean. The thing about this, when we project creations of our imagination into that deep ocean, then it becomes a mirror to us, in a sense, and how we perceive it, how we imagine it's like, and so on. So that's that famous quote from Nietzsche. So what worries me, when it comes to factual science communication, if you present a lot of information about the deep ocean to people, what I find audiences tend to do is they will latch on to individual pieces of information that resonate with what they think the deep ocean is about. Their own values, expression of identity, their own preconceptions. And it's actually really, and so consequently, it's kind of like we all see what we want to see, which is a quote from James Cameron's movie, The Abyss. I think it's very hard to share a new perspective of something, or to get people to change their perspective on the basis of new information. So that keeps me awake at night when it comes to science communication. I worry that I think quite a lot of ocean science communication is really preaching to the choir. Now, changing perspectives, though, is something I think we can achieve in a pedagogical context. So these days, I'm really proud to be looking after the deep sea ecology course that we have here in Southampton, created by Paul Tyler originally, and I've enjoyed developing it in recent years. And one of the things I get the students to do at the start of the course is to write down 3 things that come into their mind when they hear the deep sea. And these are the kind of top 5 over recent years: dark, unknown, unexplored, weird, cold. But then we repeat the exercise at the end of the module. Okay, and here are the top 5 of recent years: diverse, rich, complex, vulnerable, connected. So, it's anecdotal, it's trite perhaps, but I think it does suggest a change in perspective. So that's something I'm going to be hoping to achieve in a textbook that I've now perhaps foolishly agreed to write with my very good colleague over many years, Adrian Glover, at the Natural History Museum. We're producing a new deep sea biology textbook for Routledge, which should be ready in 18 months or so, perhaps. And we will be, I hope, very much keeping in the spirit of Gage and Tyler, 1991. But we're now 35 years on. There is more deep sea biology to be putting in there, but very much keeping with a lot of the themes there. And also, that kind of getting the facts across, that kind of fact-based perspective is, of course, a theme in engaging with policymakers, which I've done over the years around some of this research when opportunities have arisen at UK level. And then I am very pleased to be involved a little bit in DOSI, which you heard about earlier. This is the Deep Ocean Stewardship Initiative. This is a network of now more than 4,000 researchers around the world working in deep sea environments, environmental scientists, biologists, also technologists, engineers, economists, lawyers, anything that relates to the deep ocean stewardship. And it's coordinated here in SOES by Maria Baker, you can see here, with Chris Barrio-Frogen there. And I'm currently part of the advisory board for that. And the idea is to make sure that the latest findings from deep sea ecology are reaching policymakers at different levels whenever they're considering a decision that has some relevance or effect on the deep ocean. So it's kind of a science diplomacy kind of effort. And I will admit, sometimes it feels a little bit like this.
I remember when we used to get forums.
And that is, I think, the big change over the past 25 years that I've been talking to you about in this field. When I started out, it was fundamental research. It was kind of pure exploration. That is still there. But now there is this context, this strong driver to understand our impacts, to make more informed choices for the future. And that's been the big change, okay, I think, in this field. Now, over that same time, my own personal perspective has also changed inevitably because of other things that happen in our lives. So I've been part-time actually for the past 13 years. Like many colleagues, I became a carer for my parents in the last years of their life. At the same time, I became a parent, and my daughter is also different with specialist care needs. And any society is only as good as how it treats its more vulnerable members. And when you become an advocate for one of those more vulnerable members, you realise how far our society still has to go. So that is now my focus, and that has changed my perspective in science as well. So there are still lots of exciting research questions out there in the deep ocean. There are still gaps on the map. There are still exciting puzzle pieces to find. Within the picture we now have, there's that finer pattern for us to understand about why some species conform and some species deviate to that puzzle piece pattern and so on. But for me now, the big questions in ocean exploration are no longer scientific. Instead, they are about how we explore the deep ocean and for whose benefit. Because the deep ocean has now become the purview of the ultra-wealthy. And it is seductive that this could offer, greater access to us as scientists to do our work. But do we want a future where we need to become courtiers to oligarchs, providing a fig leaf for them as we use their possessions to do our research? Personally, I don't think the end justifies the means. Going to sea is rarer for me with the priorities I now have. And that makes me think about who else isn't on our research ships and how we can remove barriers to participation so that anyone, anywhere can contribute to this science and bring the benefits of a wider range of perspectives to exploring this unknown. It sometimes feels like we can't swim against the tides of history, but I think the choice of how we do our science is the one thing we can do as scientists towards building the kind of society we want to see for the future. So for me, that's now the mission. Thank you very much.
Thank you, John. I knew it would be amazing and entertaining. Really fantastic. Thank you. We've got time for questions, so I open it to the audience. Anyone has any questions?
Hi, this is
We've got one down here in front. Sorry.
Yeah, so I guess this is a very sort of pure ecological question, but something that really strikes me about the video of the little Rhinoceros and all the little crabs and stuff is that they're really active and they're really jostling around for position. Is that just a matter of trying to get into the best spot to farm bacteria, or are they being disturbed by the ROVs? Are they normally a bit more chill than that, or what's the deal?
That's a really good question. Thank you, Jethro. We always have to acknowledge the possibility that our observation may be disturbing them in some way. There are things we can do to minimise that. We can observe them with red lighting, which a lot of things, of course, can't detect in the deep sea, so we wouldn't expect that disturbance. I think it's a bit of both. With the Hoff crabs, the differences we see morphologically between the males and females aren't just about body size. The males are also kind of armoured in the claws. And when you see the video, I think Nikolai Roteman, who's really leading on that research, there's an indication that there is some sort of interaction. There may be mate guarding. There may be some form of, there's all the ecological interactions we're familiar with up here are also taking place down there. It's just where do we have the observations to realise that?
I'm not a biologist, but I was wondering about DNA. You didn't mention DNA at all, and I just wondered how much work had been done on that of these different species and your different jigsaws.
Well, it's
and to see how they related there, rather than just a straight taxonomy.
Well, exactly. So the DNA is a big part now. That's how we describe new species. So yes, we look at the morphology in detail. We look for things that distinguish them from other species. But at the same time, yes, sequencing their DNA, looking at different markers which resolve differences at different scales. So there's one very common one we call the barcode of life. We can use that, to distinguish different species in many groups. Then there are other markers that we can use where it's the same species, but the populations at the different islands, we can see how well connected they are across that range.
Right.
So it's an absolutely vital tool that we use in this research.
Right. And that is what has explained what you showed us up on the screen there. Those interconnections. That's interesting. Thank you.
Lisa.
Hi, also not a biologist, as you know. But I was interested in just looking at your map that you were sort of starting to put together, different locations, and thinking about, well, where have you all got to in terms of the competing controls, such as the tectonic setting, the geology, the chemistry, the temperature, oceanographic links, water depth, where do people think things sit in terms of what the overall controls are? Obviously, it's interactions, but in terms of the organisms and the community you get.
I think what we can say so far is all those different controls operate, but they operate in different regions. So there are some places where the boundaries are tectonic setting or history, crucially, where there was a connection and there isn't today, and so on, and isolation. In other cases, it's oceanography. And again, with the genetic evidence, we can often, we can make some estimates of divergence times in lineages, and we can often tie that into, there was an oceanographic change in this region, which meant it's now cut off from this other and now they've diverged at this level. So it is all of the above actually operating.
Okay.
Right, John. In your younger days, I remember being inspired by you speculating about life beneath the surface on our outer planet moons. Are people still worrying about that?
Thanks, Ken. It's still an area that people are excited about. So yes, I mean, there is this evidence of subsurface liquid oceans on some of the moons of Jupiter and Saturn, because of volcanic heating and particularly tidal heating, and possibility of hydrothermal activity at the bottom of those oceans. But of course, such a huge challenge to get out there, get through the icy crust. There's no possibility of any remote control, so it's all going to be autonomous. That autonomous vehicle needs to kind of realize what it's finding and not drive over something without realizing that was alive. So there are huge challenges, and things have slowed down a bit, I think, in terms of space missions out there and NASA funding and all that kind of thing. But I know it's still an exciting ambition for a lot of people working in that area.
That's one final question then from Tim.
So one of the gaps in your jigsaw was the South Atlantic, which doesn't seem terribly remote. Research ships go there quite often. So has nobody been looking properly, or what's going on?
The Chinese have done a little bit. They have a phenomenal funded program for the next 15 years, going all the way down the Mid-Atlantic Ridge, round into the Indian Ocean, all the way up through the Indian Ocean. They have started looking at hydrothermal vents in the South Atlantic, but I think it's a very exciting, it's kind of the next puzzle piece I want to target. Of course, in terms of paleoceanography, there's some interesting bits that you could target there. There are some ridge segments with a nice variation in depth. And then that different type of vent that we found on the oceanic core complex in Cayman Trough, there are plenty of oceanic core complexes on these slow spreading ridges. And I think those types of vents are probably more common. The analogy I use is they're like the dark matter of vent biology. You wouldn't find them with traditional vent hunting techniques because they don't produce a particular plume. Okay, but we now know how to find them, and they're off axis, and they do share some species with the so there's lots of exciting questions, and a nice integrated program in the South Atlantic is the place to go.
Okay. Oh, go on then. One final question. That's right.
Thank you. I have a question about the distinct biogeographic regions that you're looking at and whether the species convergently evolved or whether we're talking about such massive timelines that species did just disperse and speciate within the ocean.
Again, I think it's a bit of both. So one thing about biogeographic problems is they are kind of a consensus decision. There are always species that deviate from that pattern. But looking at individual lineages, which people have done, in some cases, yes, it is that longer evolutionary journey that we can see. And there are some fossil deposits of these environments, so we can actually go really far back. So actually for the Hoff crab, that's a family where we have a really good picture of, there's a proto-Hoff crab, if you like, fossil from probably a cold sea, but up near Alaska, and so on. So there are some lineages where I think it's that. There are others where it is that other form of more recent, allopatric speciation.
Okay, just leaves me then to thank you all for coming this afternoon. I hope you've been entertained, and I think we should show John our appreciation for a really fantastic talk.